I want to talk to you about temperate forests. And I know Germans love their forests, and where I am from, we love our forests too. But I don't want to start here or even in Wisconsin. I want to start in Ecuador, in South America. We are at the Humboldt-University. I'm halfway through the biography of Alexander von Humboldt and enjoying it immensely. I had no idea he was so connected and involved in so many political issues as well as other kinds. And what he's famous for, among other things, is being a polymath, being interested in many different things, but being one of the first scientists to have understood the connections between human land use and human land abuse, I would say, and the lack of sustainability. He observed this in Venezuela, agricultural systems that could not be sustained. Another thing he's famous for, of course, is climbing Chimborazo, which is considered the highest mountain at the time, and in particular he's famous for this Naturgemälde idea that in fact we shouldn't just think of species and communities as discrete entities. We should think of nature rather more as an organic whole that undergoes continuous changes across altitude and longitude, or rather latitude, and that each of these places on the mountain has a distinct kind of life adapted to the conditions that exist there. You may or may not know about this recent paper revisiting that volcano and documenting, using Humboldt's original data, exactly how these elevational zones have shifted over the last 210 years. It's a remarkable achievement, and it's sobering to realize that these zones have changed dramatically in that period of time. This is the longest term study of ecological change, quantitative study that I'm aware of. And you can see that the particular zones have shifted up the mountain. Agriculture as well, agriculture now going up to here instead of ending down here. This zone here has kind of moved up to the middle of this zone up there. The limit of the glacier and the snow has shifted up several hundred meters. So I think this represents not only an exemplary study of ecological change, but also a setting for my talk, which is going to be about how other kinds of ecological change are occurring right now. My colleague Volker Radiloff at the University of Wisconsin has done a wonderful job documenting exactly land cover changes. If you go to the website associated with this paper, you can actually get decade by decade changes over the last 60 years. In the amount of urban sprawl, urban cover throughout the Midwest. And this is remarkable. It's unfortunate in a way, and let me get you positioned here. This is Chicago. This is where I'm from. This is the state of Wisconsin that I'll be talking about today. And what you see here is remarkable change, and it reflects the fact that we have land. We have much more land in the New World than we've had in the Old World, and so we've been wasteful. We've spilled across the land and used it in a profligate way. So we see massive changes in land use occurring around the globe, certainly in the temperate and in the tropical zones. We know from Humboldt's work and revisit surveys and from many other sources now the extent and significance of climate change. There are some predictions that say in the future the winter may be moving down to Missouri or Illinois, and the summers in the future may be moving down to Kansas or Arkansas. This is creating a lot of problems, changing sea levels and everything else. I don't have to tell a German audience about the dangers of Waldschäden, Waldsterben, and what nitrogen deposition is doing to the world. But we have some mysteries, and the mysteries are what I want to address. In the Bielowieza forest, not too far away from here in Poland, a resurvey study noticed that 133 species that originally existed there are now almost half disappeared. At Middlesex Fells, a park outside of Boston, Massachusetts, over a 100-year period, originally botanized by Asa Gray, lost more than a third of its species in that century. In Staten Island, well, that's New York. You can figure it's going to lose species. But in Heart's Content, Pennsylvania, this is, in fact, a virgin forest. It's the exemplary virgin forest in the eastern US. It's protected, it's untouched, and yet it has lost somewhere between 60 and 80% of its understory plant species, according to my former PhD student, Tom Rooney, and his co-author here. So notice that each one of these studies is based on only the presence and absence of species at 2 time periods. No information on abundance, not replicated across space. So you could argue that each one of these is meaningless, that it's just an anecdote, that it stands alone. But I would argue that, especially as we accumulate anecdotes, these represent something we should pay attention to. So my question for today is, what drives ecological change? We know that habitat loss and fragmentation are at the top of the list when conservation biologists talk about threats to contemporary species. We understand the severity now of climate change and the dangers it poses. We've seen direct impacts of pollution. We know that succession affects ecological communities. We see invading exotic species that are having significant ecological impacts across the New World and some here in the Old World as well. I've spent many years studying the impacts of hyperabundant ungulate browsers, limiting tree regeneration and plant diversity. So we see all these forces, all these drivers acting simultaneously. Nobody has done the factorial experiment where they vary these independently and separately one at a time. Our world is doing the massive experiment in a very messy way. And so teasing out exactly what's causing the homogenization or McDonaldization of the world, the fact that each place is beginning to look like other places, and what's causing extinctions is a significant challenge for our field. What drivers do what, and where does that happen? So I first want to discuss some of the challenges we face in trying to study long-term ecological change. My study system are the forests in Wisconsin, and we're fortunate there because a predecessor of mine named John Curtis and his students collected a huge amount of data in the 1950s that we are now exploiting to study change, and I'll give you the results of some of that work. But I want to discuss each one of these drivers in turn to try and answer the question of which of these is important in this particular temperate forest. And I don't mean this to be a local study. I think what we find in Wisconsin is rather general and applicable to temperate zones around the world. And finally, I'll end up discussing a little bit about our current and future research. A clear difficulty we have in trying to understand ecological change is the fact that we do not have any consistent systems in place in any country that I know of to monitor ecological change over time. You can find abundant data on meteorological statistics, starting with Humboldt and many other people. You can hear on the nightly news exactly what's happening to the economic indicators, the Dow Jones average or the Standard & Poor's 500. And yet you don't hear anything about an ecological degradation index on the evening news. And it's a change in general that's occurring slowly enough that most people ignore it. But without the data, we don't know what's actually going on. We have what ecologists refer to as the missing baseline problem. Without baseline data, we don't know how much has changed or how much has been lost. Most of our studies are the term of a PhD thesis or a grant, and they don't extend over this window of, say, 5 years to 200 years, which my colleague John Magnuson refers to as the invisible present. Invisible because we don't have the baseline data and because the changes are occurring too slowly for most people to notice. Even worse, in a policy arena, the absence of data on ecological degradation and change is used to often imply that there isn't anything to worry about, that we don't need to concern ourselves with the impacts. This is the motivation I had for editing this book. I called it The Vanishing Present. It's a Geschenk, a present to us to have the natural world that we have, but it's vanishing before our eyes. So how should we monitor ecosystems? I mentioned that those studies of individual sites, looking at individual species and how some disappeared through time. You clearly have an anecdote if you look at one species at one site at 2 times. If you look at many sites at 2 time periods and study one species, you can really begin to answer the question, is that species declining or under threat? If you have many species that you look at one site over time, like those studies I cited, then you have multiple trends and perhaps a pattern you can look at. But the real power for ecologists come when we can study multiple species at multiple sites over longer periods of time. And this is why I've metamorphosed into a community ecologist myself. Another challenge we face in trying to study natural systems is that the things that we worry the most about, the rare species, are inherently difficult to find and study reliably using statistics. This particular species occurred at a frequency of about half of 1%. And you can see its distribution, where you can look at how often we found it or not in our sites in 1950, and how often we found it or not in our sites in 2000. And it looks as though many of the sites that did not have this species in 1950 were colonized by 2000. But of course, we have different sample sizes here. The baseline data were based on 20 1-meter square quadrats. Our own resurveys were based on 120 1-meter square quadrats. And if you use the Poisson distribution, you can calculate the probability that you will miss a species that's actually present in 20 quadrats. And it turns out the probability for a species at that frequency is almost 60%. It can be there and you will not see it. A much larger sample is, of course, much more likely to see it. But this would not be a problem if most of the species we studied were common. The difficulty comes from the fact that most of the species in most of our communities are rare. They in fact form the majority of the species we study. So we need large sample sizes. Back in the 1950s, they weren't aware of this. They didn't think in terms of statistics in quite the same way, although they invented many of the statistical sampling methods we now use. Many people look at the distribution of relative abundance of different species within one site. We have also begun to look at the relative abundance of one species across many sites using our data. The most abundant species in northern Wisconsin in the 1950s was Myanthemum canadense, wild lily of the valley. It remains the most common species. This is it down here. In the 2000s. So we can actually look at change in terms of these distributions of individual species across sites and how they have changed through time. And to summarize, a lot of what we found is that the already abundant and widespread species have become even more abundant, whereas the rare and scarce species, especially those limited to a few sites, have been the ones that have declined in general. So what motivates me to look at these systems in detail is to try and use the power of multiple species at multiple sites to understand the forces threatening biodiversity. Classically, if ecologists have always looked at local site conditions, they've asked how does soil fertility or acidity or texture, or how does the amount of light at a site, or how does elevation, how does climate at that site affect the species that are present? This is what motivated Humboldt. This is what motivated early plant ecologists in Europe and North America. But the question I want to leave in your mind is whether the world is changing in such a way that local site factors are becoming less important and that landscapes and landscape processes are becoming more important. Here is my laboratory, the forests of north and south Wisconsin. Notice that these are very different areas. In northern Wisconsin, the matrix is forested. And this is not a primary forest, a virgin forest by any means. It was all cut over in the late 1800s and early 20th century. This was a biological holocaust that occurred up here. Many species went extinct of animals. Many plant species became much scarcer. In the southern part of the state, we have agriculture and urbanization, especially in the southeast. We have the cities of Milwaukee here, Chicago's down here, Madison is there. In the southwestern part of the state, it's a mixture of forest and agricultural areas, reflecting the rolling hills of this area that's unglaciated. There's no glacial drift, and we're referring to the last Wisconsin glaciation here. The baseline data we use in Wisconsin were collected by John Curtis and his students through the 1940s and 1950s. Curtis is famous for this book, The Vegetation of Wisconsin, still in print, as well as for his idea that plant species responded individually to changes in environmental conditions, much I think like how Alexander von Humboldt imagined plant communities to be structured. Bless his heart, he was fastidious and stored all of his data in file folders in this room in my building. It took me about 20 years to wake up to the significance of what we had there and the opportunity we had to use this gold mine of data to now understand ecological change. Another ecologist who was working on the same questions at the same time at Cornell University is Robert Whitaker. And Curtis and Whitaker together related these plant distributions to soil, succession, ecological gradients. They developed new statistical methods of multivariate analysis. You may have heard of Bray-Curtis distance or Bray-Curtis ordination. And along with Humboldt, they emphasized the fact that these plants were responding to local site conditions. Site factors determine the ecological community. But in the 1960s and 70s, a more dynamic view of ecology began to emerge as well, pioneered by the efforts of Robert MacArthur and Ed Wilson, who developed the theory of island biogeography, which emphasized the fact that community composition is not static, but rather represents dynamic processes of immigration, of extinction, of turnover. And they predicted that there would be relationships between the amount of area of a habitat and the number of species that existed there. This led in turn to work by Jared Diamond on assembly rules in the New Guinea fruit pigeons and other communities, and eventually Steve Hubbell's neutral theory around the year 2000, along with Graham Bell, to try and understand plant communities and animal communities in terms of these dynamic processes. So the legacy data that we are using consists of data for hundreds of species across hundreds of sites over 50 or 60 years. So we have potentially a finer scale instrument, a high resolution microscope or telescope to look at these changes, macroscope maybe we should call it. The first thing we should pay attention to is how many of those sites still exist. And we were saddened to realize that many of these sites that were surveyed in the 1950s no longer exist. 5 out of 67 sites in northern Wisconsin had become potato fields or campgrounds or developments of one kind or another. 16 of 137 upland forest sites in southern Wisconsin have been lost to development or farming, and 3 of 53 lowland sites. So this is a statistic we should pay attention to as well, the loss of sites. We only resurveyed intact sites that had not experienced any significant disturbance over the previous 50 years. We are interested in what happened at undisturbed sites, although disturbance is clearly important. This is eine kleine Witzling. They did not actually sample parts of pavement. So what drives ecological change? The first factor that many ecologists have been studying for more than a century is ecological succession. The fact that the plants present at a site modify conditions, generally increasing shade, adding to soil, organic matter, and so forth, in a way that favors other species over time. The successional clock is reset when a major disturbance occurs. Those disturbances vary these days from development or agriculture, but originally in many parts of, especially southern Wisconsin, prairies, open grasslands dominated where fires occurred regularly. What happens now is that these prairies fall into more or less 2 camps. Some of them that are recurrently burned, and some of them that have not been burned, which are beginning to be overgrown by woody plants, shrubs, and trees. And that's important because it turns out species are adapted to the open conditions, and the regular fire disturbance are generally not adapted to the conditions that occur following succession without fire. And 2 studies are quite noteworthy here. Leach and Givnish, in a paper in Science in 1997, documented the changes between 1950 and 1987. And what they documented was significant losses of species, significant colonization by more shade-tolerant species, and a threat to these prairie systems. Amy Alstead resurveyed these prairies in 2016, a shorter period of time, but the rate of colonization and extinction has accelerated, more than doubling in the case of extinctions just in the past 20 or 30 years. And furthermore, the rate of extinctions has gone up in response to the amount of time since a fire. Without fire, we lose the prairie species. So these systems are falling apart because they've lost their disturbance regime. Check, we know succession is important. Number 2 on our list was climate change. Climate change has been studied in detail. We have the IPCC global change models from many climatographers, the downscaled models for Wisconsin, and some of the best work in North America was first done in Wisconsin. These models are now being extended to all of eastern US, show that historically we've already experienced significant changes in temperature and precipitation, and projections into the future call for more changes. My student Jeremy Ash and our group have been working on tracking, not projecting models into the future, but asking over the last 50 years, have plant species, in the same way they have changed elevation on a volcano in Ecuador since Humboldt's time, just in the last 50 years have we seen changes in the distribution of plant species in Wisconsin. We're looking at 110 species, and what you can do is you can actually place a centroid of a species distribution based on all those samples and its abundance at each site. You can create a center of mass or a sort of centroid that indicates where that species occurred in 1950s, and then you can also find where that species center of mass is when those areas were resurveyed in the 2000s, and you can connect those 2 points with a vector. And of course, you're close to the border up here, so the length of the vectors there is somewhat restricted. But if you look at all these vectors together, you'll notice the overwhelming tendency is for species to be moving to the north or northwest. Well, you might think, well, that could be for lots of reasons. Maybe they're just developing more down here or something and destroying populations. So the next thing Jeremy did was to ask, how has the climate changed for each of these species? You can ask, given its abundance and its distribution at dozens of sites in the 1950s, what was the climate that that species experienced at those sites at that time? And you can get a point in space that represents that climate, or matching it to the geographic centroid. And then you can ask, how has the climate changed based on these climate analogs over the last 50 years? And you see that that pattern also shows an overwhelming movement towards the northwest. So we can see that species have changed in one direction, climates have changed in more or less the same direction, and if you take the dot product between those two vectors, you have a measure of association that shows that there is also statistically a highly significant association between the changes in climate and the changes in plant distribution that we've observed. Again, historical. This has already occurred. It's not hypothetical. So that's a finding of interest and importance. PNAS turned it down, but we'll get it published in Global Change Biology, I think. But what was even more interesting to me is that the degree to which species can keep up with climate change varies. Some species are matching the rate of climate change with their own changes in distribution. But most species are lagging behind. They have not made it, they're not keeping up with the rate of climate change. So we see here the climate change has already affected Wisconsin plants, and that will likely affect them more in the future, especially if this line keeps changing and they're not able to keep up. The third factor, and this is one I want to spend more time on, is land use change, because it's so important and because it has some of the more interesting and significant results, I think, associated with it. I mentioned that before European settlement, this part of southern Wisconsin was mostly a mosaic: prairie, savanna, oak hickory forest, quercus. Fires occurred fairly regularly, not in all forests, but in many. But today, of course, we have an agricultural landscape The forests are generally smaller and more fragmented, certainly compared to northern Wisconsin. And they're managed for a variety of different uses. Here's an aerial view of Site 1100 in the 1950s, an air photo. That's the site right there in the center. It was sampled by Curtis and his students. We found the same site in the 2000s. It was intact, it's not disturbed, but the context, the ecology of what's going on around that site has changed completely over the last 50 years. So we took time and effort to tap into several databases to estimate road density, housing density, patch size, and a shape index within 5 kilometers. We actually did 2, 3, and 5 kilometers around each one of our sites. We also took advantage of the Wiskland data to look at forest cover, urban cover, agricultural cover on several categories. And we put these data together into analyses to try and understand if some of these changes that we were observing in community composition could be related to these landscape effects. The answer is yes. First of all, just the big picture changes are that these sites are losing diversity. On average, 80% of sites lost their diversity. On average, it was 25% at the 1 square meter level. At the site level, we were limited to matching the amount of survey work they did in the 50s, generally 20 square meters. And that was also 20 to 25%. Even more interesting to me is the fact that individual quadrats within a site now resemble each other much more than they did 50 years ago. And the different sites also resemble each other much more than they did in the 1950s. So this is McDonaldization, this is homogenization that's going on in our world. What can we learn about which sites lost species the most and what species declined the most? Let's first look at the losers. These losers are in general the pretty wildflowers. They're the kind of plants that botanists and naturalists like to find underfoot. And the winners, in contrast, tend to be already, as I mentioned to you, common and widespread species. They include this widespread herb, Geranium maculatum. It includes this vine. Parthenocissus is in the Vitaceae. It turns out vines and lianas have been increasing in a remarkable way, not only in the temperate zone, but in tropical forests around the world. Some people think it's related to carbon dioxide levels in the atmosphere increasing and maybe global warming. Other groups that increased massively in our plots were exotic invasive species like Alliaria petiolata, a crucifer, and buckthorn, Rhamnus cathartica. We decided to tease apart the two components of abundance. You can look at the abundance of any species as being the product of the number of sites that it occupies and the abundance it occurs at within the sites that it does occupy. If you take a look at these results, you'll see that there's generally a correlation. This is always true, that the species that are most widespread often tend to be abundant within sites as well. You can also see that there's autocorrelation. The number of sites occupied in the 2000s was highly associated with the number of sites occupied in the 1950s. These are standardized partial regression coefficients, or beta values, and this is a path analysis that I'm showing you. And it's actually based on logarithmic values so that we can add together the effect of sites occupied and mean frequency when presence to understand what affects total abundance at the end. You can see that actually local abundance has slightly more of an effect than number of sites occupied. But to me, the most interesting result on this particular analysis is that the number of sites a species occupied 50 years ago influences its local abundance in the 2000s, but not vice versa. Its local abundance 50 years ago has no effect on the number of sites it occupies in the 2000s. Something is uncoupled there. Something is changing. This I consider to be one of our more significant results, and it bothered me that the editors insisted on taking this graph out of the paper when we published these results. This is a species area curve of the kind that we've seen hundreds of in the last several decades. In the 1950s, there was only a very slight connection between species richness and the patch size of the forest. And yet by the 2000s, this relationship has become much stronger and steeper because of the loss of species at the smaller sites. Exactly as predicted by island biogeography, we're relaxing down to a new equilibrium with lower diversity where the sites are smaller. So this means we can't look at sites in isolation. It's not enough to protect a local site. We have to consider landscape context. We also don't know how much further this line will become even steeper due to continuing extinctions into the future. Are declines in diversity driven by land use, by urbanization? And the answer is yes. We have good evidence here that the sites that have lost diversity the most have generally had the most urban development around them. This might account for why that park outside of Boston, Middlesex Fells, lost so much diversity. We also can look at road density within 5 kilometers and ask about the active processes that maintain diversity, in particular, the colonization of native species into that site. And you can see that recolonization is a very active process when road densities are low, but as sites become smaller and more surrounded by development, this active recolonization process loses its effectiveness because of these act as barriers. Many of these plant species are limited in their powers of dispersal. And an ant-dispersed species, for example, can't get across an interstate highway. We also see that those sites that have the most forest within 5 kilometers have the most apparent colonization, the most active restocking of their diversity going on. So we see that maintaining diversity is an active process. We cannot assume that protecting a small area will suffice to maintain its diversity. To me, it was also interesting to see that this topic that's interested Alexander von Humboldt and Whitaker and Curtis so much, this issue of how plants are adapted to local site conditions, has weakened appreciably over the last 50 years. If you ask about connections between shrubs, herbs, and trees in a given forest, these are all very tightly connected to each other in the 1950s. And by the 2000s, these things have weakened a lot, such that the parts of the forest are becoming uncoupled from each other and uncoupled from local site conditions. At the same time, these surrounding landscape features, land use, land cover, have become much more important, more than doubling in significance here. So environmental similarity is less important, but geography and landscapes have become more important. We're in the world now of MacArthur and Wilson. We're leaving behind the world of Humboldt and Whitaker and Curtis. The fourth factor we looked at is invading exotic species. I mentioned that these are 2 of the 3 most abundant invading exotics. They've greatly increased in frequency, invading many of our stands, greatly increased in abundance. And again, it turns out the best predictor for which forest stands are going to be invaded by these exotic species is urbanization in the surrounding landscape. Local site conditions, like moisture index, is relatively weak by comparison. So landscape context, again, is turning out to be the story here. Well, you might say, some people argue that invasive exotic species are only passengers on this ecological tidal wave of change, not really drivers of change. So we've looked at in some detail the associations at both the site level and inside individual 1 meter square quadrats to ask whether these 3 invasives have had any effect on the native species. And what we find is that, first of all, the negative associations are more common than the positive associations, suggesting that they are having impacts. And furthermore, those negative associations are more common in urbanized landscapes, and they're more negative with exactly those species we're concerned with as conservation biologists, the rare habitat specialists. So 4 down, 2 to go. I think we're almost there. I wanted to now turn our attention to northern Wisconsin. Remember, this is the more intact landscape. Not only are the sites individually intact as required in our resampling, but now the landscape looks in better shape. And yet, even in this intact landscape with intact sites that have not been disturbed over the last 50 years, we've lost between 15 and 20% of our plant diversity at the site level. No change at the regional level, no change within a square meter, but these sites are losing species nevertheless. Who are the winners over the last 50 years? The winners are weedy, invasive, exotic species. They are graminoids, carex, poa. They're ferns like Aetherium and Dryopteris. And it's an interesting plant here, Erycema triphyllum, Araceae, known for having calcium oxalate crystals in its leaves that irritate the membranes of an animal that might try to eat it. What are the losers? The losers are Carl von Linné's favorite flower, Linnea borealis, and in general, these other pretty wildflowers, the blue-bead lily, clintonia, orchids, lobeliads. So again, we can ask, what does this tell us? Which species are being lost, or which ones are winning? Does that tell us what the driver is? And what sites are being lost, are losing the most in diversity? It's an irony, a sad irony, that the 3 state parks specifically set aside to protect the biological diversity they contained have lost more plant diversity than any other land type over the last 50 years, over half of their plant species. And what's perhaps also interesting and relevant here is that there have been no net declines in plant diversity in the Indian reservations, the Menominee, the Lac du Flambeau, the Lac-Coudray reservations, nor have there been declines in plant diversity on several of the Apostle Islands in Lake Superior. So you tell me, what's driving these changes, given these patterns of species and sites? Can anybody guess? Well, we have some implications here. Those 3 state parks that lost so much diversity have had no hunting for a period of several decades. The remote islands in the Lake Superior were far enough away not to have any deer on them. The Indians like to eat deer and act as a natural predator to contain their numbers to a density that's a half or a third or a quarter of what it is in the lands around them. So what we think is important here is that in this case, a native herbivore, an ungulate, the white-tailed deer, has increased greatly in abundance since the 1950s. This is the target deer density by the Wisconsin Department of Natural Resources, but they've lost their ability to control the herd. We have a herd of about 1.7 million right now. That is many times what we think the density was before European settlement. We can see that unhunted sites lost much less of their diversity, or much more of their diversity than the hunted sites. The species with their growing points right on the ground surface, their meristems, like grasses and sedges, can tolerate repeated cropping without dying, whereas a pretty wildflower with the meristems up high dies by being eaten regularly by deer. So why are deer so abundant, you might ask? Aldo Leopold, who founded our own Department of Landscape of Wildlife Biology, it was called Game Management at that time, pointed out that the way to manage game is to manage habitat. He pointed out that in particular game species like grouse and deer often thrive where there are grassy openings and there's young early successional habitat. It turns out this pretty much defines northern Wisconsin right now. Most of the forest is being managed for paper and pulp production, which keeps it in an early successional stage. Aspen used to be 2 or 3% of the landscape. It's now 25% of our national forest, 35% of our state forests, 45 to 60% of industrial private forest lands. So deer now have an ideal habitat. In addition, people feed deer a lot. Winters have become less severe. So for a variety of reasons, we've gone from a situation where deer used to be relatively scarce on the landscape because of Indians and wolves and cougar, and because they're not giraffes and they couldn't eat the foliage that's high up in the trees. They had relatively small impacts on the forest understory. But now we have a landscape where clear cuts are common. Early successional forests and grassy openings give them lots to eat. Predation has been eliminated or highly reduced. And so we've spent a lot of time and effort trying to understand the downstream impacts of these high deer populations on the biological systems. One way to study those, of course, is to track seedling survival, the demography of trees over time. We're just finishing a 20-year experiment where we planted hemlock seedlings in and outside fenced exclosures and have tracked their survivorship and growth. Since then, you get an idea of what those results might look like. We've devised new indices of browse. We've compared islands with and without deer, a natural experiment. And work by my former student, Katie Frecker, and others, looked at several of these exclosures in our region and asked which species are more abundant inside the fence than they are outside the fence, which ones are sensitive to deer, and which ones thrive in the presence of deer. And you can see that many species show huge differences in abundance, whether or not they're exposed to ambient deer densities in northern Wisconsin. But you might say, well, that's just one or a few places. How do we know that these deer effects really might be driving those, regional effects that we documented in our 50-year studies across the entire region? And so we did the correlation. We asked, how is the difference across the fence in a species abundance related to changes in that species abundance across the entire region over the last 50 years? And for the species in common between these two data sets, we found a pretty strong correlation. And this is remarkable in ecological studies, to find that just one factor can account for so much of the variation in the changes that we've seen. Well, what's the future of the forests if they maintain high deer densities? To understand that, you might go to the Allegheny Plateau of Pennsylvania, which has had densities of deer like what we are now have in northern Wisconsin for the last 40 years. And what they've discovered there is that the forest is being eliminated. There's no tree that can regenerate. There's no tree that can make it into the canopy anymore, which means that the forest is converting into a savanna. The understory is all graminoids and some ferns. And what's interesting to me about this particular condition in Pennsylvania is that this is exactly what Aldo Leopold found in Germany in 1935 when he spent a year here looking at the forests and the wildlife conditions here. Leopold concluded, he came back a changed man and began to speak out against the dangers of deer overabundance in the 1940s in Wisconsin, to no apparent effect. But he pointed out that the Germans had reached a point where their management for forests, especially conifers, was incompatible with game. It didn't support healthy game populations. And the management of game at these high deer densities was incompatible with forestry because it did not allow the regeneration of many of those species. So now we're on to our last factor, pollution. Is nitrogen deposition affecting plant diversity? I was one of many people associated with an effort to assess at a national level the effects of nitrogen deposition in North America. You are much farther along in this research here in Germany and in Europe than we are in the United States. But what we found was interesting. We found that plant diversity is greatly affected by what we could call terrestrial eutrophication. The higher the nitrogen, it might help at very low levels of nitrogen deposition to have a little more. That might have the effect of increasing plant diversity. But once you get above 8 or 10 kilograms per hectare per year, plant diversity falls off precipitously. And this is because plants that are extravagant in their use of nitrogen grow quickly and shade out and dominate and competitively exclude other plant species. In my own data set, I was, by the way, the skeptic in this group. I said, I did not expect in Wisconsin, we don't have situations like in Germany where you have 18 or 20 or 30 kilograms per hectare per year. I thought there won't be enough nitrogen, there won't be enough variation within our state to show any effect of nitrogen deposition. My own data proved me wrong. Yes, there's an effect of glaciation. Yes, there's an effect of hunting. But this effect of nitrogen in our own data within Wisconsin showed that nitrogen has strong and, in fact, accelerating nonlinear effects to reduce plant diversity. So we're near the end now. What drives ecological change in Wisconsin forest plant communities and by implication, temperate forests around the world? Is it succession, or is it climate change? Is it land use change, exotic species, deer, or pollution? And of course, I've shown you evidence that each and every one of these drivers is not just a little important, but very important, at least in some places and sometimes, in terms of driving declines in plant diversity in Wisconsin. So do all of these threaten plant diversity? The answer is yes, clearly they can. But what we're still trying to tease out now is how their relative importance varies among these different communities and in different contexts. The examples I gave you included the prairies and the fire-dependent communities like pine barrens are being threatened by losing their disturbance regimes. Climate change appears to be affecting plant distributions throughout the state. Land use changes are particularly significant in the southeast, where we have urbanization. Exotic species are commoner now in the south, but they're moving north and will probably have big impacts in the future in northern Wisconsin, which continues to develop itself. Overabundant deer have our data. Our most extensive data are for northern forests, but they seem to be having effects also in southern Wisconsin. Aerial nitrogen deposition is generally worse near Chicago and Milwaukee from industrial sources, and reduced nitrogen in the form of ammonium from agricultural sources coming out of Iowa to the southwest. So to answer my motivating questions, is community ecology useful or predictive? I found that it has been remarkably useful in ways I didn't expect it to initially. How can we best sustain biodiversity? I would argue that we gain a lot when we have a window into long-term change. If not for the Curtis data in these forests in Wisconsin, we would have absolutely no idea at all that Streptopus roseus has decreased 80% in abundance over the last 50 years. No one would be aware of that. But we are aware now. We know what has happened. So at least we have data where we can document the extent of change, the kinds of change, and begin to relate it to the drivers. Fragmentation, nitrogen deposition, overabundant deer, these are not having minor or temporary effects. They're having pervasive effects across landscapes now. Our current research with support from the National Science Foundation, the Dimensions of Biodiversity project, is now attempting to go. We've looked at how species are sorting into communities. We've done this sort of thing up here. But now we're completing a phylogeny, the first complete molecular phylogeny for any state in the United States, 2,400 species. We just had a talk last night. We're going to extend that to the whole eastern U.S., we think. We're beginning to look at, I showed you a little bit of results on climate conditions. We're beginning to look at genetic structure within some of these plant species to understand whether fragmentation is interrupting gene flow. Right now, I can say we've piled up a vast amount of data, and so much so that I'm inviting collaborations, because we have data that we can't publish on just because we're so busy. We have data on 28 to 35 different functional traits on 358 species. And we're using these traits to make predictions about which particular drivers might be most important. We think, for example, deer herbivory has a signature in terms of which traits we expect to change. Nitrogen deposition has a signature. We can make these predictions. And so we can look at the actual change over time. We can, if we have actually have 30 traits, it's not just a two-dimensional system, but you get the idea. We can look not how these sites have changed in species space, we can ask how the sites have changed in trait space, and whether these trait changes are associated with particular drivers. If we can figure out that deer caused this kind of a change in traits, and if we can figure out that nitrogen deposition causes this change in traits, then for any given observed change, we might be able to partition or estimate the relative importance of those different factors driving the change. So we think in terms of landscapes, geography, fragmentation. that dispersal and seed size may play a very important role. If you have a highly fragmented habitats, it's only the species that are very efficient at dispersal who will probably be able to persist over the long term in these environments. Functional traits or functional diversity is, of course, related to species diversity, but they're not the same thing. There's a lot of variation or scatter around this line. So we think we need to understand each of these independently. So vielen Dank für Ihr Interesse. Thank you to these funding sources, and thanks especially to my academic predecessors, Aldo Leopold and John Curtis, and to all the people who are actually doing this work in my group. Danke.